Hi, everybody. Welcome to another Chats with Children. I hope you're safe and well. Today, I'm delighted to be joined by Gordon Hamilton, who is CEO and founder of Depixus, and he's going to be talking about their MAGNA technology, which helps reveal molecular interactions. So, good morning, Gordon. How are you?
Very well. Morning, Rizwan.
Yeah, well, it's good morning to you. Look, just in case you don't know, Gordon had the jab this week with the booster jab. So I'm glad he's joining us today because I know when I had it, I was under the weather for quite a few days. So Gordon, thank you for joining us today. And I hope you're feeling better anyway since you had the jab.
Much better. Yeah.
Good. And now, where are you calling us in from?
So I'm sitting here in Paris, which is where Depixus is headquartered. We have a small satellite also just outside Cambridge in Harston, but this is home for me.
And good. Well, I'm in Surrey, and it's very cloudy here in Surrey today. So hopefully it's sunnier in Paris because it's horrible in Surrey this morning.
Well, I don't know. Winters in Paris are much the same as winters in the UK. It's pretty grey and dreary here as well.
All right. Well, right now, before we start talking about the Magna, perhaps you could give viewers who are not familiar with the business a little bit about an overview of Depixus and also your own background.
Well, so in terms of, I'll start with my background. So I'm initially a medic by training. Set up a first company in medical education when I was a medical student. I really just absolutely fell in love with entrepreneurship and was never able to close the doors on that world. So I worked for a while as a doctor, but then decided to leave and go into the world of biotech. So I've been working in biotech since 2005. Initially in drug development, moved out to the Bay Area in California and had a fantastic time working for a company out there called Chemocentrics. And then more recently in life science tools with Depixus. So in terms of introducing Depixus, we're French headquartered life sciences tools company. As I say, primarily based here in Paris. Team of today roughly around 50 staff, and working on development of a technology which is really about opening up a new way of looking at the biological world, particularly the world of genetics and genetic regulation and control. Very exciting adventure to have launched and to be playing a part in.
Fantastic. So let's talk about the technology you have developed. So what is it and what areas does it address in relation to pharma?
So the technology, the central piece of the technology is an instrument system. It's an instrument system. It's a tool that sits as a box on a lab bench, but in terms of a little bit for those of you familiar with the world of sequencing, DNA and RNA sequencing, the technology goes much further than the box itself. We have to be able to take a sample, prepare that sample in a certain way so that it's then ready to go into the instrument system. We have to have a set of reagents which will then analyze the sample. And then, of course, all the software to treat the data that's coming off the instrument system and ultimately convert it back into usable results for the end user. So, we're really working as a company across all that spectrum, right from the beginning, the starting sample through to the end result. But as I say, the heart and the meat of the technology is in the instrumentation piece, which is a new way of analyzing and looking at biological molecules, which we can certainly talk about in more detail.
Sure. So how do researchers currently go through that process then? Because obviously, you've got this technology, but there must be technologies out there people have already been using and methodology people have been using to already do that process.
So to kind of answer that question, I probably need to go into bit more detail on the technology and on what it's doing and how it works to begin with. Otherwise, it's rather sort of hard to talk about how it's differentiated from other technologies. So we're trying to get as close to biology in its purest form as possible. We're trying to take molecules from their cells of origin in their original native state, prepare them for analysis, put them into our instrument system, and then analyze them at a so-called single molecule level. So we're looking at individual biological molecules, and those could be molecules of DNA, could be molecules of RNA, they could be molecules of protein, and importantly, looking at how those molecules interact with other molecules. And I know that our conversation is probably going to focus largely on the pharma piece, and it's in those interactions where we feel we can start to reveal things with more detail and clarity than is possible before. So once the sample is prepared and put into our instrument system, we're then capturing those molecules as individual molecules, and we are then repeatedly interrogating those molecules. We have a way of being able to look at those molecules, to flow binders into a flow cell that interact with those molecules. And those interactions we can pick up, we can detect those interactions in our system, and we can look at the strength of those interactions. We can look at the way that different molecules bind, how they bind, where they bind on these individual molecules of interest. And we can therefore reveal a lot of information about how different molecular entities interact with each other. I'll just, I should give that a slightly more concrete example. So we're particularly excited in looking, for example, at the world of RNA today.
Yeah,
so,
sure.
RNA is, as I'm sure you're aware, it's a highly structured molecule. Unlike DNA, which has this rather standard double helix form, RNA can, as a single-stranded molecule, is not just a single linear filament. It's really often forms multi-complex structures, secondary or tertiary structures that bind and fold together in a way which is, in many ways akin to the world of protein. There are these motifs, these intramolecular hairpins that have these very clear secondary and tertiary structures, and that's a target for drug development today. There's a lot of excitement about RNA as a drug target.
Sure.
And we have the way to capture RNA molecules in this native state, in their folded state, and we can then introduce molecules into our flow cell that would bind to these RNA molecules, particularly binding to particular features, particularly secondary or tertiary structures, and we can reveal how those molecules are binding, where they're binding, and the strength of those binding interactions. And the obvious kind of next step in that is, I'm sure you're imagining, is that if those molecules happen to be small molecules that are being considered for drug development, that becomes very interesting, because we can then start to reveal how small molecules' potential therapeutics are binding to their targets, whether they're stabilizing those RNA secondary structures, whether they're destabilizing them, and how strong those interactions are, and how strong that stabilization or destabilization is. So it's really looking at molecules in a very new and very different way. So in terms of what other people will be doing, using today, obviously there's a lot of Mass Spec that's used for looking at these sort of interactions. Much less dynamic. There's SPR, surface plasmon resonance, which is widely used as well. Slightly closer to the sort of dynamic information that we can provide, but much more limited in terms of the richness of information that can come out of the system. And then there are other technologies, but perhaps those are the two that are worth highlighting the most in terms of being roughly or a little bit analogous to what we're doing.
Right, so it's exciting.
Does that help answer your question a little bit? I don't know whether that's...
No, it has done it. So it's pretty much you've developed a new technology out there then to look at these molecules in a way that currently is being looked at, but in a very different way. I suppose that's the right way of saying it. Right. Okay.
Really very novel, and there is a sort of an emerging world of single molecule sort of pharmacology, pharmacodynamics, which is we think we're right at the beginning of what could be really very, very transformative for the whole field, for the whole drug development field, and much more broadly than just RNA alone.
Right. Well, I mean, one question comes to mind though is that obviously you said people are already using Mass Spec or SPR technologies to do this though. So although yours can do this, why would they use yours rather than theirs? Then that would be the obvious question.
I think because we can get a lot closer to the true reality of binding, of understanding the way that molecular entities are binding to each other in a really dynamic sense. This capacity to repeatedly analyze what is happening, to build up a kinetic picture right from often, for example, KD is obviously very, very important in the world of medicinal chemistry, and those KDs are often.
Give my ignorance. What's a KD?
It's the dissociation kinetics of a drug binding to its target, and we're building up a picture of these dissociation kinetics based on the kinetics of the on rate and the off rate of binding. We're really getting to actually seeing, to essentially almost visualizing in real time the binding of a molecular entity to its target and how strongly it's binding and its release. And I'll give you an example. It's perhaps not direct into the pharmacological world, but in terms of detection of base modifications, or base modifications, things like methylations that are occurring on DNA and RNA. We're detecting base modifications on these molecules by looking at antibodies binding to their target. So, for example, an antibody raised against 5-methylcytosine will bind against its 5-methylcytosine target, and with our system, we can see the binding of that antibody, that single antibody, to its target, and we can see the moment on a computer screen in front of our eyes when that single antibody is releasing itself from its antigen, from the 5-methylcytosine. That's giving us a real-time view of kinetics of what is going on in that interaction and the strength of that interaction. And we can repeat that analysis over and over again. We can build up this very, very detailed picture of what is happening at a really very fundamental level, which is really quite different to what other technologies are capable of doing, which tend to be much more in bulk assays where you derive information from very large data sets. Here again, each and every molecule we're analyzing is its own individual experiment, if you like, where we can start to analyze what is going on that individual molecule, and then that can be done in parallel across multiple, tens, hundreds, thousands of molecules. So we can gather data, but as I said, each and every molecule is its own independent experiment where we're gathering data. So that's really build a picture.
No, it does, certainly. And you said it's also used within other industries or other markets as well. So what other areas can you also use this technology, or are you targeting with this technology?
Well, so the background of this technology comes out of, that was spun out of a biophysics lab. So this is, at its base, this is a technology which is known as magnetic force spectroscopy, or the instrument system is often referred to as magnetic tweezers. So it is, at least in the biophysics community, the technology is reasonably widely known and reasonably widely used for studying, for understanding the biophysical properties of biological molecules. That's where it comes from originally, is trying to understand, how the building blocks of our life are put together. So we've, as I say, spun out of a biophysics lab to commercialize the technology, and for us, we're looking to commercialize, at least initially. It's the technology itself. I mean, the term is often misused or rather loosely used. There's this notion of being a platform technology. I hear it bandied about all the time, but I think that term is very, very appropriate for our technology. This is a technology we've got a wide diversity of different potential applications. For what we're interested in terms of the early applications for the technology, the sort of things we want to be working on together with our early customers, is going to be in two areas. The first area is in the area of drug development, as we've just talked about in a certain amount of detail. The other area is in the area of nucleic acid analysis.
Right.
So more akin to the world of genetic analysis around sequencing but an extension of sequencing, starting to look at parts of the genome which are very difficult to analyze with current technology, which are typically focused around next generation sequences largely, Illumina sequences and others. what we're really interested in that application space is revealing, again, aspects of the genetic world, the genomic world, which are difficult to reveal. So things like base modifications on DNA, base modifications on RNA, so the whole epigenetic world, and also the world of interactomics, so how nucleic acids are interacting with other entities in the cell, with transcription factors, RNA binding proteins, and so on. One avenue to pursue with that is in the area of diagnostics, is in tissue and liquid biopsy. It's a very, very hot field today, as you're probably aware. Being able to take a blood sample and analyze the self-free circulating DNA in that blood sample. And all of the companies that are operating in the space, I think bar none, to my knowledge, are looking not only at genetic features, but are looking at epigenetic features. It's a really crucial part of these assays, is being able to reveal epigenetic patterning. And that's just not all that straightforward today. And we think with MAGNA, we've got some very, very significant competitive advantages in being able to reveal not only the genetic aspects of from tissue and liquid biopsy, but also the epigenetic world, which gives a lot of additional positive predictive value in these tests. So we hope we're going to be a technology which will be really, really important in the tissue and liquid biopsy space, as well as in the drug development space.
Brilliant, Gordon. Well, thank you for giving that overview. So someone who's watching this now, there are a couple of questions will come to mind, I think. One is it available now? So where can I get it? And secondly, where can I find out more? So is it available currently? And if not, when do you think it will be available? And where can they get more information?
So we're working pretty hard at the moment to bring this to market. So not commercially available today. So we've just, as you may have seen, just closed our Series A financing just a couple of days ago.
Yeah, congratulations.
Thank you. Really very, very happy about that, and excited for the next chapter in the company. And it's quite a big shift in the company. So we've been previously working sort of more focused on the research phase of the business, proving what the capabilities of the technology are, and being able to generate some robust data sets to prove that out and getting that published. And now we're shifting our focus to the commercialization, to the commercial development of the instrument system, with the aim of ultimately getting it to market. And our hope is that it'll be on the market, at least through an early access program, within the next 18 to 24 months from now. That's very much our aim. Start to get it into the hands of key labs around the world that are wrestling with some of these challenges that we've just been talking about, keen to trial and try out new technologies to be able to reveal these aspects of the dynamic genome, for want of a better term, is sort of to look at genetic regulation and control, who are interested in understanding and be able to reveal more linked to tissue and liquid biopsy, and also in the pharmaceutical development world as well.
Great. And so if people do want to get more information, then how can I get more information? Do they just go to the website?
Our website. That's and there's a really nice animation. I'm sorry, I'm a little biased, but I think it's a really nice animation which will give viewers an idea as to how the technology works at a pretty fundamental level. It's 3 or 4 minutes long, well worth a watch. There's also a link to a paper that we published in the Nature journal Communications Biology not so long ago, to dig into more scientific detail. And of course, then there are ways to be able to contact us as a company, and to for any of your viewers who might be keen to potentially collaborate with us. We're starting now to open up early collaborations with labs that are excited by the sort of subject areas we've been talking about, and we're certainly delighted to start to make more connections across the world in these spaces, and start to get our technology into other people's hands, not just our own hands, and used and trialed, and help us to develop it and ultimately bring it successfully to market. So.
Well, fantastic, Gordon. Well, look, there you go, viewers. If you'd like to collaborate with Gordon and the team in terms of getting this a new technology out to market, then please get in touch with Gordon and the team via the website or through LinkedIn. There'll be lots of ways to communicate to them and get in touch. So Gordon, all that stuff is, well, first of all, congratulations on the funding. That's fantastic news. Well done on getting the booster as well. Hope you feel better. Yeah, and good luck with the next phase of your journey with the business as well. I hope it all goes well over the next few months in terms of getting people using and collaborating with you. So, all that's to say is I wish you a fantastic, happy, healthy, and wonderful festive season and New Year. It's been lovely to meet you and learn more about Magna as well.
You too. Very, very much. Wishing you much the same. Yeah.
Thank you very much. And viewers, there you go. I hope you found that interesting. If you'd like to know more about the technology and to collaborate with Depixus, then please get in touch with Gordon, go to the website, find out more as well. The link for the website will be above the video. And all that stuff, when he says, I wish you a fantastic festive season. Stay happy, healthy. Until next time, stay well. Bye-bye.